In the human cell, the nucleus is the protected chamber that holds genetic instructions. To keep this environment stable, the cell relies on a network of quality control machinery that removes damaged components and maintains order. In ALS, that order begins to break down. ALS is a disease where motor neurons, the cells that control voluntary movement, gradually deteriorate. For decades, researchers have tried to understand why these neurons fail. A recent study published in Neuron by scientists at Baylor College of Medicine points to an unexpected culprit, a protective protein that becomes too aggressive for the cell's own good. The protein is called volosin-containing protein, or VCP. Under normal conditions, VCP is essential. It identifies proteins that are damaged or misfolded and helps remove them before they accumulate. This process is so fundamental that VCP is found across nearly all forms of life. But in certain genetic forms of ALS, mutations push VCP into overdrive. Instead of selectively removing faulty proteins, the mutated version begins targeting structures the cell depends on, including parts of the system that controls movement in and out of the nucleus. The nuclear pore complex is the system that regulates what enters and exits the nucleus. It ensures that genetic instructions, regulatory molecules, and essential proteins move in and out with precision. When VCP becomes hyperactive, it starts dismantling parts of this gateway that should remain intact. Over time, the nucleus loses its structural stability, and communication between the nucleus and the rest of the cell begins to fail. This breakdown has a direct impact on another protein deeply linked to ALS, TDP-43. In healthy neurons, TDP-43 stays inside the nucleus, where it helps manage RNA processing and gene regulation. But when the nuclear pores are compromised, TDP-43 can no longer remain where it belongs. The cell is hit from both sides, the nucleus loses a critical regulatory protein, and the cytoplasm becomes burdened with toxic clumps. What makes this discovery even more important is that VCP-related dysfunction isn't limited to one rare mutation. Variants of VCP have been linked to multiple neurodegenerative conditions, including forms of ALS, frontotemporal dementia, and other inherited neurological disorders. That means the mechanism uncovered in this study may represent a broader vulnerability in the nervous system. To explore potential solutions, the researchers used cell models, human-derived neurons, and fruit flies engineered to carry disease-linked VCP mutations. When they reduced VCP activity in the fly models, levels of nuclear pore proteins stabilized, and the animals showed significant improvements in movement. This shift from trying to replace lost functions to dialing back an overactive system represents a meaningful change in how scientists might approach treatment.